cancer cells actually are replicating. They provide all the nutrients necessary for a virus to have good, robust infection, as well as cancers avoid the immune system. So these are all good aspects in general about viruses and tumors. However, viruses also use pathways that are activated selectively in cancer cells, and that's why you can engineer viruses to make them selective. But viruses that naturally occur also use pathways that are activated. And so we were interested in how does this virus selectively kill just these types of tumor cells but not normal cells, and how do they exist in nature. So this is a picornavirus. It's a single-stranded RNA virus that the first step is essentially it's a very small particle, 20 nanometers, which gives it an advantage in terms of getting into tumors and spreading and getting through the fenestrations in the bloodstream. So it actually has to bind to the cell, and then it actually gets uncoated, and the RNA actually gets translated to make structural proteins, as well as the virus actually replicates, and then the virus is assembled, and then it's extruded from the cell, and the cell essentially bursts, and that's actually one of the mechanisms how these viruses kill cells. So we're interested in where in this whole pathway is the selectivity of why some cells are killed and why some cells aren't. And so we actually started to do the following experiments. So we actually labeled the dye an Alexa-4 dye, and in this picture actually you can see the virus actually throughout the cytoplasm. This is the nucleus. And so what we did is actually screen both cells that are killed by the virus as well as cells that are not killed by the virus. We were just asking the question, is it possible that only when the cells are killed by the virus does the virus get into the cells, and where cells are not killed, do they not get in? And that would suggest that the selectivity might be at the attachment step and essentially getting into the cell. And that's exactly what we found. In fact, the majority of the non-permissive cells essentially look just like this, where the virus wasn't getting in. The majority of the permissive cells look like this. So the next thing we did is we took a permissive cell, we actually put it into a mouse, we made a hybridoma. We made monoclonal antibodies, believing that those monoclonal antibodies could be screened to block the ability of the virus to actually bind to the cell. So we got two monoclonal antibodies called AG10 and 1711, and these antibodies had very interesting features. And so for instance, this is a control antibody. 1711 here completely blocks the ability of the virus to bind to the cell, whereas this antibody, AG10, doesn't block the ability of the virus to bind, it blocks the ability of the virus to internalize. So you can see it actually lining up on the cell membrane. So then we actually made a library, a lentiviral library from the permissive line. We actually put it into a non-permissive cell, in this case CHOK1. So this is a Western blot using this antibody AG10. And if you look at a permissive cell, you can see this pattern here. And if you look at this non-permissive CHOK1, you see this pattern. So then we put the library in, and what we see actually is we found several clones that had a similar pattern to what you see in permissive human cells. So we're in the process now of identifying the gene in that lentivirus to be able to identify at least what is the component necessary to internalize this virus. So we believe that the majority of the mechanism is involved in the attachment and internalization. And so another aspect that we did at the same time was to actually look at permissive lines and non-permissive lines, doing gene expression arrays, as well as mass spec bioinformatics on cell structural proteins. And we found actually this pattern here, this set of genes that are all structural genes, actually predicts quite well whether or not these cells are sensitive or not. One interesting gene that I'm going to talk about is integrin alpha-4. So you can see integrin alpha-4 very well predicts actually whether or not the cell actually is permissive or not to a particular virus. And interestingly, several of the genes that were there actually turned up in different stem cell sets, as you can see here. And one of the things that was done by one of our collaborators at Baylor College of Medicine was actually to demonstrate that this virus can actually select cancer stem cells, at least from medulloblastoma. And we think that's an interesting part of what this virus may actually do, both in cancer cells as well as possibly in nature, and why it actually has remained hidden so long and doesn't actually cause disease. So I mentioned integrin alpha-4. So this is actually an example of integrin alpha-4 here. And so we actually did a couple experiments to determine whether or not integrin alpha-4 was involved. And one of the first things we did is we got the crystal structure of the virus shown here, and we found that there's an LDV in amino acid motif known to bind integrin alpha-4 beta-1, and this was on an exposed loop of the virus, so it was quite possible that this bound alpha-4 beta-1. So we can mutate this specifically, and when we mutated it, we found that we could not rescue the virus, again suggesting that that particular motif was actually very important. So a few other experiments we did was we took a cell line that didn't have integrin alpha-4, K562, versus one that did. And when you infect these cells, you can actually measure viral production over time. And this is the background cell here, K562, so you actually see some background virus replication occurring here. However, and this is actually the wild type, a fully permissive line per C6. And you can see when you put integrin alpha-4 in here, you actually make at least 1,000-fold higher levels of virus than you do without integrin alpha-4, again suggesting it was involved. We also see an increase in binding with another cell line where we put integrin alpha-4, for instance, into a CHOK1 cell line, although it's certainly not as high as a fully permissive small cell lung cancer line. So this data, as well as some other data that we've obtained, strongly suggest that integrin alpha-4 is at least one essential component. And to make the story a little bit more complicated, it turns out to be that it needs to be activated form, and it's activated in small cell lung cancer by CXCR4. So again, it looks like similar to what's seen with other viruses, there's a pathway that's activated in cancer. So now I'm going to spend the rest of the talk concentrating on our phase 1 study. With this data, we went into a phase 1, and we concentrated on tumors with neuroendocrine differentiation in adults. Patients had to be positive for one of 3 neuroendocrine markers, such as chromogranin A, synaptophysin, or NCAM. We looked at various neuroendocrine tumors, primarily carcinoid cancers. We did actually do some small cell lung cancer patients. It was all single intravenous dose administration. It was actually done in an outpatient setting, so the patient went in for a couple hours, got a 1-hour IV dose in 100 mils of saline, and then was monitored for another hour or 2, and then essentially went home. It was a 3+3 dose escalation in log increments, so you could do, you did 3 patients before you had to go to the next cohort, unless there was some safety issue, then you could do a 3 more. We start at a low dose, 10 to the 7th vector particles per kilogram. We wanted to go at least 10,000-fold higher to 10 to the 11th vector particles per kilogram, or dose-limiting toxicity. We believed that 10 to 11 vector particles per kilogram would be a dose that would be effective in the majority of the cancers that we were trying to treat. We also monitored viral kinetics and clearance in every single patient. So basically we were taking nasal swabs, sputum, blood, urine, and stool, and we were measuring virus, both by looking at viral titer, as well as genomes and PCR analysis. So we did this essentially for all 30 patients. And we also looked at neutralizing antibodies in these patients. Excuse me. So 30 patients were dosed predominantly with carcinoid, small cell lung cancer, and various neuroendocrine tumors. Every single patient in the trial was actively progressing by RECIST criteria. And what we saw was, excuse me, no dose-limiting toxicity, no drug-related serious adverse events, and any drug-related AEs were simply mild, transient flu-like symptoms. The result for the small group of small cell lung cancer patients was that we had a 3-fold overall improvement in survival as compared to historical controls. So I'm actually going to go through the small cell lung cancer data in a minute. And as well as the carcinoid patients had approximately a 70% response rate, which included PRs by PET, near PRs by CT, and stable disease. We had about 12 sites. The main study sites were Johns Hopkins, Mary Crowley, in Texas, Leahy Clinic, as well as US Oncology had 8 small sites throughout the US, as well as Mount Sinai. So carcinoid cancer is actually an interesting cancer. It's a slow-growing cancer with a primary site that generally is in the small intestinal or terminal ileum. And you get regional spread of all carcinoids in lymph nodes and then the liver. The incidence is pretty low, and there's only 8,400 patients per year that are newly diagnosed.